Hello guys, the topic of today is formation and differentiation of primary germ layers. What is gastrulation? Gastrulation is a process involving a complex series of cell shape changes and cell movements occur inside the blastula, or we can say that the formation of blastula to gastrula is known as gastrulation. And when the blastula is converted into gastrula, there is so much changes in the cell, like the formation of three primary germs layers, and there is a cell movement known as epiboly and emboli, and the that three germ layers which are formed during gastrulation, they are ectoderm, mesoderm, and endoderm. These three layers are the basically layers which give rise to human germ layers. Ectoderm is exterior, mesoderm is middle, and endoderm is inner. These three layers are ectoderm, mesoderm, and endoderm. Ectoderm is ectoderm मतलब the layer of outer like hair skins, okay, and the organ which formed on ectoderm is brain, okay, spinal cord, and internal ear. These are the layer formed from ectoderm. And from mesoderm, like cardiac cells, bone marrow, blood and lymphatic system, gonads. And from endo मतलब inner layers are formed from it, like urinary bladder, thyroid, parathyroid, thymus, etc. from it. And the blastocyst have the inner cell mass of cell. Actually, what is happening, blastocysts floating in the uterine cavity, day 5th. You can see in the photograph the day 5th blastocyst floating in uterine cavity. In day 6th, blastocyst adhere to the uterine wall. This blastocyst adhere मतलब stick to the uterine wall. The outer layer is tropoblast, and this outer layer stick and the inner cell mass. Actually, this tropoblast give rise to placenta and any other functions, and this inner cell mass give rise to embryo. This tropoblast furtherly convert into chorionic villi, data primary chorionic villi, and give rise to placental function, and the inner cell mass give rise to embryo. Okay, the implantation begin as trophoblast invades into uterine cavity. As trophoblast begins uh, invades into uterine cavity, the implantation occur, and during implantation many hormones are secreted like scg, which maintains the cell shape and release the amount and increase the amount of actual progesterone. Blastocyst, you have to be seated the blastula, they having the two layers, tropoblast and inner cell mass. Okay, tropoblast and inner cell mass. Outer layer give rise to placenta. That is, the placenta is the connection between mother, and it is a organ which connect mother to child, mother to embryo, so that the exchange of matter take place, and the inner cell mass give rise to embryo. And the next one is, in the first step of implantation, trophoblast cells proliferate and form two different layers, an inner layer or cytotrophoblast. Trophoblast form two different layers, an inner layer or cytotrophoblast. When cell proliferation occur outer layer, or the outer layer is known as a syncytotrophoblast, where cells lose their plasma membrane. In syncytotrophoblast, cells lose their plasma membrane. Cells lose their plasma membrane and fuse into the multinuclear mass of cytoplasm. The syncytotrophoblast project inversely into endometrium and digest the uterine walls in contact. So what's happen in it? When cell proliferation occur, when implantation occur, trophoblast, we have a trophoblast that is break down into two layers. First is being cytotrophoblast, that is inner layer, and the outer layer is syncytotrophoblast. Actually, you can see suggest Thought it is a uterus and implantation occur. The trophoblast layer is being this, and from this two layers are being formed. The outer layer is known as the syncytotrophoblast and the inner layer is cytotrophoblast. That's okay. The syncytotrophoblast projects inversely into the endometrium of mother, endometrium of uterus, and adjusts the uterine cells as it is in contact. So you have to be see what happened after implantation. The trophoblast of blastula convert into proliferate, form into two layers, differentiate into two layers, inner layer and outer layer. Inner layer is known as cytotrophoblast and outer layer is known as the syncytotrophoblast. And you have to be see that the endometrium line of uterus and how the blastula, meaning how this get attached with the and how this get attached with the trophoblast. Trophoblast is the outer layer of the cell. This is attached with the uterine cavity and form the inner layer. The inner layer that is known as cytotrophoblast and outer layer that is known as the syncytotrophoblast. You have to see that the endometrial stroma with blood vessels and glands and two layers are formed. It is known as cytotrophoblast and syncytotrophoblast. This is another lumen of uterus. And you have to be see that the day 9 implantation continue. Inner cell mass form bilaminar disc. The inner cell mass form bilaminar disc like structure. In the day 11 implantation complete, amniotic sac and yolk sac form. The embryo about day 9 after fertilization, the embryo inner cell mass has now divided into two sheets of cell known as epiblast and columnar. Epiblast the columnar cells and hypoblast the cuboidal cells. Together the epiblast and hypoblast make the bilaminar two-layer embryonic disc. The sac formed by the extension of epiblast is the amniotic sac. The sac formed by the extension of epiblast is known as the amniotic sac. The outer membrane of the amniotic sac is amnioid. The yolks are formed by the extension of hypoblast. You have to be see that the trophoblast here and the inner cell mass, the amniotic cavity, formation of yolk sac, the ectoderm and the endoderm. The ectoderm, the primitive stick in between arise, which give rise to three layers. And next is wing, the primitive stick and the three-germ layer. The three-germ layer begins to form on 14-15 day when a raised groove called the primitive sticks appear on the dorsal surface of the epiblast by invagination of the inner cell mass or emboli, the amnion, the primitive streak, primitive groove, epiblast. You have to be seen here. You seen here in this photograph amnion, primitive streak, primitive groove, epiblast, mesoderm, hypoblast undergoing replacement, endoderm formation of primary germ layers are formed. And the next is being primitive streak. The epiblast cell migrates inward at this streak. On this streak, epiblast cell migrate towards inside towards this streak on days 14-15. The first cell that migrate through the primitive streak displace the cell of the underlying hypoblast to become the endoderm. The primitive streak, epiblast, inner layer is hypoblast, and inside the cell is endoderm. On then starting on day 16, the ingressing epiblast cell form a new layer between epiblast and endoderm. Then on starting day 16, two layers are formed that is known as mesoderm. The epiblast cell that remain on the embryo torso will make up the ectoderm. The epiblast cell that migrate through the primitive node towards the notochord. Fate maps in early embryos. Meaning of fate map. A fate map is a diagram of an egg. It is a diagram of egg or blastula indicate the fate of each cell or region at a later stage of development. Fate maps are essential tool in most embryological experiment. Actually, in most embryological experiments, fate map is the essential tool. They provide researcher with information on which portion of the embryo, actually on which portion of the embryo will normally become which larva, on which portion of the embryo which come which larval or adult structure. Actually, which embryo give rise to this structure or which layer give rise to this structure. This is fate map. The analysis of fate map on each blastomere after first and second cleavage is called cytogeny. Construction of fate map. Fate map is of different types of animals have been constructed by following methods: observing living embryos by natural marking in some invertebrates, the embryo being transparent and having relatively few daughter cells. They remain close to one another. They have possible look It has been possible to look through the microscope and trace the descent of a particular cell to the organ by subsequently form. The type of study found by the Edwin G. Klein in 1905 in that initiates stela paritata where the different cell contain different pigment. For example, muscle forming cells always have a yellow color. Vital dye marking. Most embryos, however, does not have the facilities. Transparent, few cells, different colors are described by the Stella Perdita. It was in 1929 that Vogt was able to trace the fate of different areas of amphibian eggs by applying fate of different areas of amphibian egg by determined by applying vital dyes. This vital dyes stains the cells without killing, which have no effect on the development of the embryo. Dyes are Nile blue, natural red, Bismarck brown, etc. Radioactive labeling and fluorescence dye. A variation of the dye marking technique is to make one area of the embryo radioactive. A donor embryo is taken and grown in a solution containing donor embryo. We have to take a donor embryo and grow in a solution containing radioactive thymidine. The thymidine base is subsequently incorporated in the DNA of dividing embryo. A second embryo acting as the host embryo is grown under normal condition. The region of the intercut from the host embryo and replaced by radioactive graft from the donor embryo. The cells that are radioactive will be descendants of the cells that are graft distinguished by the autobiography. So, genetic marking is another one method. Radioactive and vital marking. Radioactive and vital dye marking have their own drawbacks such as dilution, our many cell division, and the laborious preparation of slides. One permanent way of cell marking is to create mosaic embryos having different genetic constitution. The best example of such a marking is graft girl cells inside a chick embryo. By doing so, fine structure of maps of chick brain and skeletal system can be made. Usefulness. The fate map of organism helpful in tracing the morphogenetic movement of the cell and the ultimate position they take up in future. This is very helpful for how which layer will give rise to ectoderm, which give rise to mesoderm, and which give rise to endoderm, which will form blood forming tissue, which is where heart form, and which layer give rise to heart. This is very helpful. Thank you guys.